First, we have the Dean of MTSU's College of Basic and Applied Sciences, Dr. Bud Fisher.
Good morning. It's my job to welcome you today on behalf of the President, Dr. Sidney McPhee, and the Provost, Brad Bartell. When I was coming over here, I was thinking about science. I started a long time ago. And I would trade places with you in a split second today to be back there in my career. But something has changed hugely since I got here. And one of that is that the first class I ever went to at Syracuse University, which was a biology class, had 3 women in it. And today I stand in front of 350 young ladies interested in science. Probably the greatest revolution that's occurred in science has nothing to do with molecular biology, has nothing to do with what would be microbiology, has to do with the fact that women have taken over the sciences. It has literally changed the way we do things, and it has brought way brighter people than us males into the field. And all I do today is challenge you to go out, have some fun today, and find your passion in science. And once you find that passion, I'm going to tell you, stick with it because you'll find an incredibly rewarding career. And with that, have a great day.
Next, I would like to introduce Ashley Gatlin from Nissan North America. And I'm going to tell you a little bit about her real quick. Ashley Gatlin is a senior manager for Nissan North America Incorporated in the Supply Chain Management Department of the Smyrna Manufacturing Plant, where she manages a high-performing team responsible for the manufacturing plant bill of materials and other supply chain functions. Gatlin has also led the Nissan Smyrna Manufacturing Product Quality Assurance Inspection, Warranty, and ISO departments, and the Pre-Delivery Repair Department. During her management, the ISO 9001 team led the effort to achieve the first site-wide ISO 9001 2008 compliance for the Nissan North America Incorporated Smyrna Manufacturing Plant. Gatlin has participated in the Art to STEM, which is, we all know STEM is science, technology, engineering, and math, and the WISDEM, Women in Science activities supported and led by MTSU and Nissan North America Incorporated Smyrna Manufacturing Plant, where she has encouraged young ladies about the importance of choosing STEM careers. She has received the Nissan Vice Chairman Ambassador Award, as well as the Total Customer Satisfaction Quality Award for work in certain areas of vehicle. Since 2007, Gatlin has been a member on the Vanderbilt University Department of Mechanical Engineering External Advisory Committee and was the co-founder for the joint Vanderbilt-Nissan program for providing real-world engineering design projects for senior engineering students. Please give her a hand.
Oh my goodness. Good morning. How are y'all doing? Y'all excited to be here? Awesome. I'm really excited to be here, and I'm quite honestly blown away by the number of young ladies here today. So my name is Ashley Gatlin. I work for Nissan North America. On behalf of Nissan North America, I would like to thank you for attending today and encourage you to continue to explore your horizons in math and in science. And I guess 2 quick points along those lines. As you know and as you've heard, these areas, math and science, they can quite literally open any door for you as you continue growing up, going through middle school, high school, and college. Makeup design, chemistry, aerospace design, my personal favorite, car manufacturing and car development. The sky is the limit, but here's the thing. If those doors are going to open up for you, you have to do what you're doing today, buckle down, study, and you're going to have to persevere and be very resilient as you go through those careers. As we heard earlier from the dean, not so long ago, there weren't a lot of women in science. You really didn't hear about it. I graduated 1998 from Vanderbilt, which doesn't seem like long ago, but it really was to me. There were 4 other women in my major in mechanical engineering, and right now I'm a member on the advisory board for mechanical engineering at Vanderbilt, and that number has not increased so much. So I would like to see you stay where you are and continue learning and going in through these fields so we can have more young ladies and young women in these careers. So that's my first point, is stick with it, be there, and the sky is truly the limit. The next point, and this is not so related to math and science, but it's just something I want you to think about through the day and as you go on. Have an open mind and be willing to try new things and to do new things. And I think today it's going to help kind of stretch that. But I'm going to give you an example. When I was getting close to graduation, my senior year in college, we had a really, really hard class. It was really hard for me. It's called heat transfer. Well, I didn't think I was going to make it. It was so tough. I made it, had some great professors, buckled down, graduated, and I was so glad that class was over. And as I got out into the workforce and I'm paying back my student loans, doing all that, one of my first jobs was washing cars. And I'm thinking, about 10 o'clock at night, I've been there for about 12 hours, longer than that, I'm washing this car. And I'm thinking, why did I struggle through heat transfer and pay back? Why am I working to pay back this student loan? But then it just kind of clicked. Wow, I have a job at this wonderful company, and I have so many opportunities in my future. And I was like, this is pretty cool. I'm getting to wash this car that nobody has ever seen. It's not even been revealed in a car magazine yet. And it just kind of clicked for me. That's pretty cool. And over the years now, I've been able to take advantage of some of those opportunities at a wonderful company like Nissan, and I'm looking forward to the future and moving along. So I guess that's what I'd leave you with, is be open to new things like that. And if someone says, all right, Miss Hotshot Engineer, just graduated from Vanderbilt, I need you to go wash those 10 cars. Okay, I'll do it. No problem. So that's what I'd like to encourage y'all to do, and have fun today. Really glad to have you, and thank you for the opportunity to talk to you. I guess with Dr. Gross, she and I talked a little bit before. Do y'all have any questions? Anybody like to ask anything about engineering or anything at all? Yes, ma'am.
Can you tell everybody what kind of math you needed to study and how they can prepare for it as middle school girls and high school girls?
Yes, I can. I did not get enough math in, so by the time I got to engineering school, it was a little bit hard to kind of get everything started. Algebra 2, all kinds of algebra. Just get that where you're not even thinking about it. You're just doing it automatically. Geometry. Everything kind of in life comes together in shapes, geometry, and if you think about it, everything's a math problem. You're getting in your car, you're going to drive to school. You kind of have you have to solve a problem on how you're going to get there. So you're thinking geometry, algebra, trigonometry needs to be like this. And if you have the opportunity to take calculus in high school, absolutely do it. It will lay a wonderful foundation for you, whether you're going to study biology, chemistry, engineering. Try to get all of those math classes in. And I would also encourage you, don't do it alone. Work in study groups. And as soon as you start to have a question or a concern, raise your hand and start asking those questions, because very quickly, as y'all know, with math, if you start getting behind, it kind of builds and it snowballs. So raise your hand and ask the questions, but definitely algebra, geometry, trigonometry, and calculus. Get that calculus in if you can. Do y'all like calculus? Is it fun? Y'all like math? Who likes math? Do you like math? I love math. It's like a puzzle. Yeah. Okay. There is an internship program. I believe the internship program is for college age. I will need to get some detail on that. I do not have specific detail with me now. Job shadowing, I have seen some job shadowing there. My experience with it has been at the college age, but I would be happy to get that information, and then I can get that back to Dr. Gross, and then Dr. Gross, you can disperse that information. Yeah, love it, love it.
I'm Shatoria Simmons. And I'm Karma Washington. And on behalf of MTSU EYH, we would like to say thank you for coming and speaking with us this morning.
And here's a gift from us to you.
Thank you.
Okay, right now I'd like to ask all EYH sponsors to stand up and be recognized. And then all girls, give them a round of applause to tell them thank you. So now I'd like to bring Shatoria back up and also Jayla Turner, and they're going to introduce our keynote speaker for today.
Currently works at Vanderbilt University Medical Center for Health Service and Research as a project.
We're introducing Frenika Mentor, and prior to this position, she worked for the Ridge Associated University, also known as the Oak Ridge Institution for Science and Education, as a health education specialist in the health communication, technical training departments, health and safety training group, and at the Tennessee Department for Health, Heart Disease, and Stroke Prevention program as a public health educator. He has both a master's in science degree in health, physical education and recreation, with a concentration in health 2005, and a bachelor's of science degree in chemistry 2002, and from Middle Tennessee State University.
Has completed all of her coursework and is working hard on her research, studying interdisciplinaries and health service used to complete her dissertation.
All right, here is Frenika Mentor.
How many of y'all have been afraid to do anything that was new? Sorry for my southern accent, y'all. Yeah, I've been afraid to do something new myself. And I'm telling you, to do something new, you have to be brave and courageous. To do something you never did before. I believe that if everyone had that mindset to try something new and try something they've never done before, they'll realize they'll be able to do things beyond their dreams or more than they could imagine. And that they'll be able to do things they never thought that was never possible. Sometimes you have to think outside the box when you're trying to do something new, which will be uncomfortable. It feels uncomfortable when you're thinking outside the box, which leads to my next tip. Step outside the box. Step outside your comfort zone. Sometimes you have to step outside your comfort zone to explore new things. Actually, when you step outside your comfort zone, that's when the magic happens. So face your fears, be brave, suck it up, be a big girl, and leap outside your comfort zone into the unknown. So today will be a great opportunity to make that leap and to expand your horizons in research fields such as science, technology, engineering, and mathematics. The whole world revolves around those fields. So take the time today to research and explore the fields of science, technology, engineering, and mathematics. Now, I have these questions on here. So that means don't be afraid to ask questions today. Ask the who's, the what's, the where's, the when's, and the why's. Because that's the only way you're going to learn if you ask questions. Don't think just because you're asking questions, people will look down on you. Chances are they want the answer to the same question. It just took and they will admire your bravery just for asking the questions. So don't be afraid to ask questions. You probably asked a question that someone else would be wanting to know. So when you leave here today, go read up on the field and the subjects that you're interested in. That's another way that you can explore your horizons. Another thing is get connected. Get connected with those that work in the fields that you're interested in. Ask if they have any job shadowing opportunities so that you can learn what they do on a daily basis. You never know, those connections can become mentors, which leads to my next slide. Let me talk about mentoring. Mentoring is a pick to the brain, an ear to listen, and a push into the right direction. This is a quote by John Crosby. A mentor provides an informal, yet highly valuable relationship with their mentees. In this relationship, as I mentioned from the quote, mentors pick your brain. Yeah. They provide an ear to listen, and they give you a push into the right direction by sharing strategies they use for success and unwritten rules to their profession. Being mentored is a great opportunity. Talk about some benefits of having a mentor. And I have listed them on the slide. You'll get individual recognition, encouragement, and support. So you'll be with a person one-on-one. Sometimes there's like group mentoring, but I like the one-on-one mentoring because you learn more. And you have an increased self-esteem and confidence when dealing with professionals. Before I had a mentor, I was scared to speak in public. I was scared. You wouldn't even see me up here today. So just they'll give you that type of self-esteem, that boost to your self-esteem and confidence. Confidence to challenge yourself, to achieve new goals, and explore new alternatives. They give you a realistic perspective of the field, like what you need to know when you're going into something. They give you advice on how to balance work and other responsibilities, how to set priorities. They give you the knowledge of the do's and don'ts in the field. And because they have experience with others and have experience on how to get connected with others, they will give you experiences, tell you things that you need to know for networking opportunities, to get connected to other people in the field. And I can tell you, it's not all the time what you know, it's who you know to get you further. So if you know a lot, that's good, that's great, that's awesome. But share what you know with other people. Get connected with other people. Now I am going to go in how did I expand my horizons and how it expanded my opportunity. Now, how many of you all are in middle school? I see it divided in middle school. Oh, that's awesome. That's a lot of you. Well, you guys you have an early start, and that's great because the earlier you start, the better. How many of you are in high school? All righty, that's great. So now is the time for you guys to be serious. You all should be serious, but you guys have the opportunity to make the best of this conference today. And you can use all that you have or what you gained today to expand your horizons, to more, just a little bit more seriously, to expand your opportunity. With this said, I'm going to transition to how I expanded my horizons and how this expansion led to more opportunities for me. I didn't expand my horizons until I was a sophomore in high school. I went to Riverdale High School. Anybody else in here that went to Riverdale? Yay, go Warriors. This is how I expanded my horizons and how this expansion provided me with the foundations and research that I needed to prepare for my current position as a research study coordinator at Vanderbilt. Now, when I was a sophomore, I was at the end of my sophomore year, and I didn't have any intentions in going to college because on my dad's side, there were people that went to college, and I just didn't spend too much time on my dad's side of the family. But on my mom's side, that's where I spent the bulk of my time with, and they either went to the military or got a job at a factory, didn't go to expand, to college and expand their horizons, didn't expand their education. So I was thinking, okay, so when I get out of high school, I'm going straight to the military. This is my thinking. I didn't have college on my mind. So I've always made good grades, and particularly in math and science. So close to the end of my sophomore year at high school, my chemistry teacher at Riverdale, Miss Leanne Richardson, told me about a solicitation about the American Chemical Society Project Seed program and urged me to apply. Being me, the person that liked to try new things, the brave one, I applied, and I was awarded one of the 2 slots. So let me talk about how the Project Seed program helped me to expand my horizons and led to different opportunities. Well, the Project Seed program was a summer chemistry research internship for economically disadvantaged high school students. It provided opportunity for students that were historically lacking exposure to scientific careers. And in this program, you spend a summer conducting hands-on research. My first time in a lab with a lab coat and goggles, and so you can get the picture. So we worked closely with the scientists in either academia industry or government in a research lab. I also was able during this experience to try new things and explore new opportunities. So for 8 weeks, I worked with, my first year I worked with zinc. And then the second year I worked doing research on telluric acid. So for 8 weeks in the summer, I had this unique opportunity to work with a great mentor, Dr. Gross, who helped me not only to develop laboratory skills, but to adequately conduct scientific research. And it also enhanced my written and oral skills too, because I told you guys I was afraid to get up and speak in public. But after that, I had to present my research, so I had no choice to get up in front of everybody. So Dr. Gross provided me with guidance, encouragements, letters of recommendation. She actually pushed me into the direction to try something new, apply for college. So my mentoring relationship with Dr. Gross still remains today, even though I've moved on to other things that I was interested in, all the things I've tried something new. Well, I'm always trying something new. But those research skills that I got at age 17, I carry with me to this day. So before my senior year in high school, I took my ACT. Didn't know, those are old pictures. It's like Flashback Saturday or something. It's not Flashback Friday, it's Flashback Saturday. But those are old pictures. Before my senior year, I took my ACT. I applied for MTSU as a chemistry major, received a full scholarship to my amazement, and I graduated in 2002 with my bachelor's and had an opportunity to go to UT Health Science Center that I also took. I tried it. I had a mishap, life mishaps, and withdrew, but I didn't let that stop me. Despite that, I did not give up. And that's the thing that you should not, that's something you should do. If anything happens in your life that you think is a failure, it's not a failure. The only time it's a failure is when you give up. So do not give up. Keep going. Do something. If it didn't work that way, try something new. So do not give up. So I returned back to Murfreesboro and applied to MTSU again, and I was interested in health education. So I changed my focus to health education, and I applied and was accepted to the graduate program to pursue my master's in health, physical education, and recreation with a concentration of health. And I graduated with a master's degree in December of 2005. So during my undergraduate years, I was presented another opportunity that I seized, of course, and it was the Ronald E. McNair Post-Baccalaureate Program, which was a federal TRIO program that was funded by the U.S. Department of Education in a TRIO grant. So I applied for that, and the McNair program was for undergraduate students to prepare them to get a doctorate degree, to go into graduate school and succeed there. And it was geared towards first-generational college students and underrepresented groups like Black, Hispanic, and American Indian descent. So to qualify, you had to be part of those two groups, and you had to have a desire to pursue a PhD, and you had to have 60 credit hours and a 3.0, and I had all of those, and I was like, oh, I'm gonna try something new. I got brave and tried something new. So they selected 20 students, and I was one of the 20, and they provided them with a stipend and GRE preparation, a mentor-guided research opportunity. So I did research all through from high school and my undergraduate. So those research skills were expanding, which expanded my opportunity. And it also provided travel for different conferences where you present your research. So this was another way I made connections. I got to get to make connections. So I was the first McNair student at MTSU to present at a state, regional, and a federal level conference because I had the prior research as a, undergrad. Well, I can say that I am currently working on my PhD right now. And I was talking about life mishaps. There were things that happened from my master's to the point I started. It took me 6 years to start my PhD journey. But the knowledge and skills that I obtained from MTSU, from high school to MTSU, in the undergraduate and graduate levels, prepared me for this journey. So, y'all, these companies up here, these are all the companies I worked for. MTSU, Center for Health Services Research, the Tennessee Department of Mental Health and Developmental Disabilities, and the Division of Alcohol and Drug Abuse, the Tennessee Department of Health and Heart Disease and Stroke Prevention, the Oak Ridge Institute for Science and Education, which is managed by the Oak Ridge Associated Universities and the U.S. Department of Energy. And all of these things prepared me for my last opportunity, which is Vanderbilt University Medical Center, where I am now a research study coordinator for informatics studies, health informatics studies. And those health informatics studies require a lot of computer experience. I didn't get a lot of computer science experience when I was in an undergrad because I focused on chemistry, biology, and so that's another area. So if you want new experiences, explore computer science. A lot of statistics course. You need to know how to measure your, make sure that your research is measurable, and biostatistics was a good course. So if you need to, statistics is a really good way to measure things. So with those opportunities with the companies I worked for, I was able to work with other companies, 3M, like as a subcontractor, or I did work through as a contract, which is the Centers for Disease Control and Prevention, the National Library of Medicine, the National Cancer Institute, Agencies of Healthcare Research and Quality, the U.S. Department of Health and Human Services, U.S. Department of Energy, the U.S. Environmental Protection Agency, the U.S. Nuclear Regulatory Commission, the Tennessee Valley Authority, the Federal Bureau of Investigation, and the Tennessee Valley Healthcare Systems, VA. So other ways that I expanded my opportunities, and I've embedded those in this presentation as I was going, but Louis Pasteur, anybody know who Louis Pasteur is? Do you know? Yeah. Exactly. Give her a hand. That was an awesome answer. That was awesome. So he said, chance favors the prepared mind. So with that said, here is a way that you can prepare yourself and expand your opportunities. Go to networking events. This right here, this conference, expand your horizons. This is almost like a networking event. You can make friends here. My sister was at the first Expanding Your Horizons. And she met a friend, and they stay in contact every day. They're Facebook friends. They follow each other on Instagram. They call each other. You can come to my daughter's birthday party. You can come to my daughter's birthday party. And it's like they're sisters. So it's also a networking event where you can get to know other people in the field that you're interested in. Get to know some of these leaders here. They can be very good role models, team leaders, and find someone that you can get connected to. And take some educational and training courses. If you, the fact that you're here at this conference is a way that you're expanding your opportunities. If you can find some internships or job shadowing opportunities, that would be great. Get a new job or a project. I had a friend whose daughter wanted to be a veterinarian. So during the summer, this is like an indirect way to become a veterinarian, to get to know animals a little bit better. She took little bitty jobs like being a neighborhood dog walker, telling people like if you're going to be out of town, I can go feed your dogs or take care of them or your cats or whatever. So this is an indirect way to get acquainted with animals, but it was her way of saying, hey, I love animals. This is what I want to do in the future. This is the way I can do. But it was like the little small steps she's taking. So one of the ladies in the neighborhood, she got her connected with a local veterinarian. She watched her dogs and got her connected with a local veterinarian. So that's a little way to get connected. Enhance your knowledge and skills by, what was the first tip I gave you? First tip, expand your knowledge by what? Reading. Read. Not only read, like research about what you want to be, what you want to do. So you'll get to know, it won't be all new when you try to pursue it. And increase your visibility. The fact that you're here today, you're increasing your visibility. Get out in the community, work in the community, get out and work in the field, job shadow. Find some connections. So, if I can do it, you can do it. A girl that didn't think she could go to college, that didn't think she could do anything, but now she's out there. I look at my life now and I was like, how in the world did I do that? And then I thought about it and I put it all in this presentation. I was like, this is how I did it. I'm going to share it. So looking back over my life and realizing how far I come, I have to say that I didn't even know I had it in me. I tried things that were new, and I surpassed my dreams and my aspirations, and just by trying new things. So expand your horizons today, because it will expand your opportunity. Thank you.
Okay, does anybody have any questions for Fernika?
What was your favorite subject in school?
My favorite subject in school was all types of sciences, because I like the laboratory portion of it. Chemistry, because this is what urged my about the project, because I like to mix things that I was told not to mix. And one day it caused a bad thing in the lab. Let's just say that, and they had to call the fire department and everything and evacuated the building. But it wasn't an explosion. It was, enough that they had to come clean it up.
Do we have any other questions? Okay, hold on.
Were there a lot of girls in your classes?
Actually, there Well, I took a lot of honors classes. I don't know why I was taking honors classes. It was by recommendation, because if I had my choice, I wouldn't have, but it was by recommendations. The teachers that saw something in me urged me to take honors classes, and I'm glad they saw that in me to take those honors classes. It was like 3 or 4 girls in a class. But as I got into high school more and more and more, there were more girls classes. And actually in college, I was amazed at how many girls were in my class. I walked in and I was like, I'm usually surrounded by boys, you know? So there's more and more girls that are becoming interested, which is a good thing. The thing is, it's retaining those girls in and keeping their interest in science. That's the key. So we have to keep them interested.
Where do you work now?
I work at Vanderbilt University Medical Center. I work in a research lab, healthcare informatics. And that entails, what it is it's a mixture of computer science and electronic health records, and you're synthesizing data to either enhance quality in healthcare or make healthcare better.
Was there ever a period in your life where you doubted your science career?
Yes, there was. And you know those times I told you I had life mishaps and not to give up? Yeah, there were some things I thought, there was sometimes, and you'll feel like this. There's going to be some times in your life that you may feel inadequate, that this is not cut out for me, this is for boys. No, this is for you. The fact that you're here today and trying to explore and expand your horizons lets you know that this is for you. You're trying to learn something new. You're trying to get to know different fields. So, yeah, I did, but do not, when you have those kind of times in your life, do not give up. Keep going, and if it doesn't work one way, try it another way.
Is your job fun?
Yes. Yes, I am learning new things. I mean, you may think I have it all the knowledge, but I am learning new things every day. And I get to propose new things. That's the good thing about it. And people actually listen to me. I don't know why, but they do.
Okay, so can everybody give Frenika a hand?
So I went to MTSU and started undergraduate school. What sucked about it is I was 21 years old, 22 at the time actually, 22 years old, and I couldn't act like a normal 22-year-old. I couldn't go to parties. I couldn't do that. I had to come home. I had to come home to a baby, right? Time management was critical for me. I really had to push through and get those grades that I needed, and it wasn't messing around anymore. I wasn't there to have fun. I was really there to find a career for myself. I started undergraduate research with Dr. Chong here at MTSU, fell in love with research. I love research. I'm a lab rat, according to my dad. I love being in the lab. 24/7, let me be in there, as long as there's some windows, because it can get pretty dark. But I love being in the lab. So I decided to pursue that as a career, and I went ahead and got my master's. I started my master's degree here at MTSU as well. During my master's degree, my professor, my thesis advisor, Dr. Dunlap, she had a great opportunity for me to go and to go ahead and start my career up at Vanderbilt, working for Craig Lindsley for the Neuroscience Drug Discovery Center that they have there. It was great. Dr. Lindsley was really supportive of me trying to finish my master's. So I actually worked part-time before I was full-time, and I worked full-time the last semester that I finished my thesis. So if you can imagine, I'm a single mom, I'm working part-time then full-time, I'm taking classes, and I'm trying to finish my thesis research. Guys, I don't really have much of a life. Thank God I love my job. So this is a big picture of me. This is actually on the MTSU webpage. If you go to it, they've used it now for 3 graduation ceremonies. This picture, I am elated. From what I've been through in my life, this is the accumulation of everything. This is my rare emotion in that moment. I got it. I got a master's degree. And through everything that I've been through, this was it for me. This is also a picture of me in my lab up at Vanderbilt. This is me, just doing standard chemistry stuff. That's me all day long, lab coat and everything. So we're going to get started in the drug discovery process. This is, again, just reiterating to you guys the process that we're going to take you through. So first we're going to start with target selection. Where are you going to begin? What disease are you going to go after? Then we're going to talk about, we're going to put these two together, the lead discovery and medicinal chemistry. This is my world, what I do on a daily basis, and I'm calling it hit to lead process. We'll figure out what that is in just a moment. And then we're going to get into the in vitro and in vivo studies. In vitro in Latin actually means in the dish. So the in vitro studies, what's great nowadays in terms of biology, they've allowed us to take cell lines and put them in a petri dish. So we're able to see what the drug is doing in the lab before you put it in any sort of animal. But, of course, you also have to do some of those animal testing as well. And then we'll move on into clinical trials and talk about each phase of those. So let's get started. So target selection is the first step in the drug discovery process. I have right here a definition for you. Finding an agent with a particular biological action that is anticipated to have therapeutic utility. So let's break this down just a little bit. Finding an agent. So an agent can be anything from a gene to a protein to a receptor. It's going to be anything with a particular biological action. So what this means, it has to have something to do with your disease. You have to find something. We're not going to go after a gene that has something to do with breast cancer when we're trying to find an antidepressant. So we have to put the two together. It has to have anticipated therapeutic utility. We're not going to go after something that's never been proven before that it's going to work, that it's going to actually do anything in your body. You want to go after something that what we have called proof of concept studies. We'll get to that in just a moment. So now we're going to talk about some aspects with target selection. What constitutes an improved medicine? So what that can be is taking some receptor that somebody has hit before with a drug. How can you improve upon that? The other aspect to target selection is innovative research. So this is going to be when you are going after a new target that nobody's ever gone after before. You have your own intuition, your innovation about what you think is happening in that disease and how, in terms of, when I say we or they, it's a pharmaceutical company. So some strategies involved with target selection is going to be speculative research and innovative improvement. So again, this is kind of what I've touched on before. Speculative research is going to be when you're going to go after something that nobody's gone after before. That's in terms of a protein or a receptor or a gene. That is speculative research. Also, the innovative improvement. So this is when you're going to make something better. If you guys know anything about antidepressants, each one of them kind of go after a different target in the brain, dopamine or serotonin. There's lots of serotonin antidepressants out there. So when each of them came on the market differently, that was called innovative improvement. You were improving upon what was already on the market. This will make so much more sense when I get to my example, I promise. Some challenges involved with this is your disease mechanism. Guys, we are complex biological systems. I'm sure you guys already know that, right? Complex biological systems. That means that we have genes that encode for proteins. Those proteins are going to go hit a target and make something happen. Well, where is the drug going to go? How are you going to get that drug to any target protein that you want to get to? So these are some issues that you have to go through when it comes to target selection. And the last part of target selection is really the most important. Just because a protein is involved in some sort of disease does not mean it's druggable. And by druggable means you can get to it by a certain compound that we would put in your body. So just because you have a protein that encodes and gives you a certain disease doesn't mean that that's good enough in order to be a drug. So how does this relate to the first word in drug, determined? We've got a few definitions up here. having made a firm decision and being resolved not to change it. When it comes to target selection, this is really big in pharmaceutical companies. You have to make a firm decision. We're going after this target. We're going after this receptor. That's it. You have to be determined not to change it. The reason being is there's 4 or 5 steps down the road you got to go. You can't start changing things when you're 3 steps down. You have to be determined. You have to make sure that this is what you want to do. You have to be committed. You have to be firm about what you want to do. Purposeful is one of my favorite synonyms when it comes to being determined. You have to have a purpose. So how does this relate to you, right? How does this relate to you guys and being a woman in STEM? Well, you think about it in terms of what I've done through my life. I had to be determined to come back to school. I couldn't just be like, take a class, oh crap, this isn't for me, I just want to go get a full-time job, I don't want to do this anymore. I had to be purposeful. I had to have a purpose. I had a little one at home that I had to make sure had a future. How am I going to do that if I'm not purposeful and determined, insistent that I can make it? The second step in the drug discovery process, I'm terming hit to lead, all right? So this is going to relate to resilient. So how do you find a lead compound? There are 4 facets involved in finding a lead compound when it comes to drug discovery. Have you guys seen Breaking Bad? You know what Breaking Bad is? Okay, so it's about the high school chemistry teacher who starts making meth in like his house or something. I actually haven't seen it, just kind of know about it. Anyways, that's what I do on a daily basis. I do not make meth. No, I do not make meth, guys. I'm not saying that. I make drugs. Potential drugs on a daily basis. That's what I do. So you think about it in terms of where are you going to start? So there's a structure associated with meth. There's a structure associated with every compound that I make on a daily basis. So where's that structure coming from? I'm not just pulling it out of the sky like, let me make this today. I'm not doing that. There are what they call high throughput screenings, where they're going to take structures that have been made by various companies. A lot of companies will actually even sell you what they call databases full of these structures. And this high throughput screening is really cool. It takes all these structures and it's going to, what they call do a biological assay, and it's going to find its affinity for your target. Does it like your target, essentially? That's what affinity means. Does it like it? Does it want to be there? If it doesn't, then they're not going to actually show you that. When it comes out of the screening, you're going to get what they have, probably just like a summary of some of your best compounds and what we call hits. So that's when I say hit. It's a hit from the screening. Again, this is going to make so much more sense when I go through the example. A few others that we have is the fragment-based screening. So that's when you're going to take different parts of a structure and screen those, and then a medicinal chemist would come by and put those fragments together in order to make the actual lead compound. And the other one, which I'm doing actually right now, is called patent busting. Do you guys all know what patents are? So you own the rights. Well, we actually own rights to structures. Sounds kind of weird, right? But we own the rights to those structures, and that's how pharmaceutical companies make their money. They own the right to that structure, so nobody else can make it. And that's where you get generics. The patent ran out. So what I'm doing right now is called patent busting. I'm going to bust the patent right open. I'm going to say, whatever you can do, I can do better. That's what our company is saying. This company did that, we're going to make a better one, is what we're doing. And that's actually what I'm doing today on a daily basis. So lead optimization, the next kind of subset to this, is where you have to have what my Dr. Lindsley, my boss, calls a med chemist's eye. You have to be able to know what maybe your target is looking for. What kind of structure does your target want to be? And so this is when we get into library synthesis. So we don't know at first, right? Like we've got this hit, we kind of know, but we have to go through it a little bit. Library synthesis is when we're going to synthesize 10 to 50 compounds at one time. In one reaction in the lab, we are going to synthesize up to 50 things, which is great because you can get 50 different affinities at one time in order to know what you want to move forward for lead optimization. Again, talking about biological assays. Assay just means experiment, essentially, in terms of your guys' understanding. And what that means, again, is what I've kind of been saying, affinity for the target. Is it on the target? Is it off your target? How well does it want to be on your target? The other part to the biology is the metabolitic profile. Is it being metabolized? How is it being metabolized? And are those causing issues? And if all these things come together, you have a candidate drug. Now, I'm going to tell you before we move on, this whole slide right here, well, excuse me, this left side, because I haven't gotten to resilient, takes up to 5 years of work. So what I just told you in 5 minutes takes 5 years. Can you imagine? That's crazy, right? So I started my job 2 years ago. So what I started 2 years ago, I'm maybe halfway through finding what we want in order to get a candidate drug. Pretty crazy. So how does this relate to being resilient? So here's some definitions for being resilient. Able to spring back into shape after bending. To withstand or recover quickly from difficult conditions. If you guys have been listening, I'm sure you can tell where I'm going with this. Being a single mom at 21 years old, I had to come back. I had to say that I had the potential, and I had to be resilient and to get where I needed to go. This is relating to you and your guys' lives. Things are not going to go the way you want them to. They're not going to go according to plan. It's just not going to happen. This has to do with hit to lead because reactions don't work in the lab. How many of you guys are actually taking chemistry? Okay? Chemistry or already taking it. Okay, you guys set up experiments in the lab. Okay? They work every time. Do you know why they work every time? Because somebody made that curriculum like 10 years ago and knows that it's going to work. I do stuff that's not going to work every single day. It's not going to happen. You have to be resilient. You have to be able that if I want that structure, there's got to be another way to get there. That's what I've had to do in my life. I've had to be flexible, I've had to be strong, and I've had to be tough in order to get where I am today as a woman in STEM. So moving forward with preclinical testing. So this is lab and animal testing in order to determine if a drug is safe enough for human testing. This is where you put together your profile in order to go to the FDA and said, we can go into humans. Everything you guys put in your body is toxic. Okay? You have too many apples in a day, you're going to get sick, right? Tylenol. Have you guys ever read, like, the Tylenol label? You guys ever read it? If you take too much Tylenol, you're going to have renal failure. You're going to have liver failure. Everything you put in your body is a toxin. But you have to weigh the benefit versus the reward. You have to be able to understand it is toxic. How is it toxic to your body? But the benefit is greater than the risk. So in terms of preclinical testing, you have to define the spectrum of toxicity. You have to know what exactly it's doing. So in terms of this could be side effects. You have to know whether or not this is going to cause problems, and if they're problems that you can come back from. You have to determine the exposure. Are you exposed to the drug too long? Again, these are experiments that the biologists do on a daily basis in order to determine a lot of these things. Assess the potential human risk. There is risk in every drug you put in your body, but again, balance the risk versus the reward. Preclinical testing can be summarized in one sentence. What exactly is the compound, your candidate drug that we got to? What is that doing in your body? Where is it going? How are you excreting it? Is it on the target? For how long? Is it going to get to the brain in terms of what I do with neuroscience? Once it's in the brain, how's it going to get out of the brain? Things like that are very important. So how does this relate to being unwavering, being steady and firm, and having no hesitation? Again, kind of goes back to being resilient. Things are not going to go the way you want them to. And you have to set up experiments that are going to tell you what you need to know. So this goes back to being steady and firm and having no hesitation. If something is going wrong with your compound, you have to figure out how to fix it and move forward. You have to be unfaltering and relentless. As a company or as a medicinal chemist, you have to be able to say, I want this compound, we want it to work, how are we going to get it to work? And there's different aspects in doing that. You all might be thinking right now, well, if it's toxic, why am I going to want to make it work? Well, there's different avenues to being toxic. Toxic is not all on the same level. There's different levels to toxicity. We're not going to get into that right now, but there's different levels. And so even though something's necessarily toxic, does not necessarily mean it's bad for you. So unwavering. How does that relate to me and my life and my discovery as a woman in science? I had to be sustained. I had to stay the course. I had to stay focused. I I had to be unwavering. When something didn't go my way, I had to be relentless in terms of grades, in terms of job opportunities, in terms of being a mother. I had to be enduring. I had to stay the course. So lastly, clinical trials. This is kind of the most important part, if you can think about it in terms of the drug discovery process, because we're going into humans, guys. We are going into humans. This is exciting. If a company gets to phase 1 clinical trials, they've done something right. They've gotten to this point. And to this point probably took them upwards to 8 years. Again, the timeline, 8 years to get to this point. You know how long clinical trials go? I think some years are on here. You see this? Oh, not there yet. Nima Laser. 6 to 12 months. So we'll go upwards, right? Now we'll go on the downward. 6 months, 1, probably gonna be 2 years. We'll go 2 years here. So 2.5 to 3. We're going on. This is the earliest you can get through phase 3 is 4 years. 4 years after you've already put through probably 8. The drug discovery process is 12 to 15 years long. Where you guys were like born, right? Some of you guys are 15, yeah, 15, 16, okay? When you guys were born, that's a drug that came on the market last year. That's when they started that process. That's crazy. But if you relate that back to you all, it doesn't come overnight. I did not become this determined, resilient, unwavering woman overnight. That didn't happen. It's a process that's been taking effect since I was in middle school. So you guys, it can start now. So let's get into clinical trials. I went on a little side note. Phase 1 is going to start with healthy human volunteers. All 3 of those are very important. We're going into humans. They have to be healthy, and they're volunteers. So this is where you're really going to see the action of the drug. Is it even helping healthy people? Because if it's toxic, correct, like I told you, every drug is toxic, right? If it's toxic, why are you going to put it in sick people and make sick people more sick, right? So we have to see what's happening in the healthy people. This is about 100. Sometimes this can be like just 20 people, depending on the drug that you're actually going after and the target. And again, this is like a 6 to 12 month thing. Sometimes it can be 3 months. Depends on the disease and the medication that you're going for. Once you get through phase 1, so this means that you understand how you're going to dose your patients, and that you know that it's not toxic, that you balanced that risk versus reward. So we're going to get into phase 2. This is where the magic happens. The first large study is very controlled. There's obviously a placebo. This is where you're going to get into several hundred, again, volunteers. This is the first time you're really going to see what your drug does. This is probably the most exciting in terms of what I believe the most exciting phase when it comes to clinical trials, because you're finally going to see if all this hard work, possibly upwards to 10 years right now, has really paid off. Do you really have a drug that's actually going to make people better? It's actually going to help people. So this is what phase 2 is going to do. It's going to give you the drug effectiveness. Is it really effective in terms of getting to your target and making sure that you're balancing the risk versus reward? You're really going to understand side effects at this point. You're also going to understand dosages. You guys know doctors. If you go to a doctor and they're going to like, all right, we'll give you 5 milligrams of that stuff. And you come back and you're like, it's really not working that well. It's kind of working. He's like, okay, well, let's give you 10 milligrams. So what that called is dose dependency. And so you really want that with a drug. You want to make sure that as you increase the dosages, you're increasing effect. And that's going to come to fruition when it comes to phase 2. So you got through phase 2, your drug's looking great. We're going to move on to phase 3. This is where it's widespread. This is when you're going to see several thousand patients with your disease seeing effect, hopefully. And if you get through phase 3, you're going to understand what you're going to put on your label, because there will be side effects, right? We've heard those commercials like, you're going to know. Those are all the side effects, right? They say them so fast. Gosh, I really wonder who that person is. Man, they probably get paid like hardcore money, right, to just talk really fast so people don't understand the side effects. It's really funny. At least I think it's funny. So that's going to provide the label instructions. This is also going to be where this is where you're going to have your evidence. They're going to go to the FDA, and you're going to tell them, we want our drug to be approved. Big deal. This is really big. So you're in several thousand humans. and you want to get there. Okay, so how does it relate to being a girl? So a girl, definition of a girl, person's daughter, mademoiselle, a damsel, not in distress, right? We are women, we are strong, we are tough, we are not in distress, we are determined, we are resilient, we are unwavering. This is where everything comes together when it comes to clinical trials. You have to be determined that you are very confident in your drug. You have to be resilient. Things in clinical trials are not going to go the way you want them to. You're going to possibly see side effects you didn't see before when you did the cell lines in the petri dishes and you were in the animal testing. Things are going to happen. You have to be able to be resilient, bounce back from that. Being unwavering, you have to endure whatever happens. Sometimes things are not going to go right. Your drug may not make it to phase 3. It may not get out of phase 1 because of something that happened. But you have to be enduring and you have to stay the course. And you have to stay focused. You have to be a determined, resilient, unwavering woman in STEM. So how does this relate to my life? Well, I think it's in terms of everything that I've ever had to go through, right? Being pregnant at 20, having to go back to being a single mom at 21, having to go back to school at 22, having to get my bachelor's degree and a master's degree while still working part-time and full-time. I had to be all of these things. I had to make sure that I stayed the course. I had to be very determined. And when something didn't go my way, I didn't get a job, I didn't get that promotion, or, that experiment in the lab just like crapped out and now I've lost all of my material and I have to go back to 3 months ago and start making that stuff again. You have to be able to bounce back. And I want you guys, I challenge all of you to turn the me into you. So let's go to a case study where we're going to put this all together. So what I just told you guys in almost 30 minutes, like crazy, right? Blowing your mind. It would have blown my mind when I was in high school. We're going to give you a real life example so you guys can kind of see where this all fits together. So this is a drug called Servexant, and this is a hypnograph of somebody, a normal adult, just falling asleep. So this is what they wanted to be an insomnia medication, so for a sleep disorder. And this was really cool because this was innovative improvement. This is on a new receptor that had never been done before when this first started in terms of going after insomnia. So they went after the orexin receptor, again, had never been targeted before in terms of insomnia. This orexin receptor regulates your sleep and reward pathways. It's raised when you're awake, and it's depleted when you're sleeping. So what they wanted to do is they wanted to take something, get a drug, get it to the orexin receptor, and actually suppress it, right? Because it's low when you're sleeping, so they didn't want it to turn it on. They wanted to essentially turn it off and keep it off, because when it turns on, levels are elevated, that's when you're awake. So again, this is innovative research. This was something that had never been done before, but they did have proof of concept studies. They did have those studies in the literature that told them that this was a possibility, that this could really work. The company was very excited about this because they really wanted to improve on some of the problems that we're having with some of the insomnia medications. A big major one was the side effect that the next day you have locomotive problems. Depending on your dosages, you can't drive a car. Well, I need to sleep in order to drive to work, but I can't go to sleep because I can't take the medication that's going to allow me to drive. It's a problem, right? So the company was really excited about trying to work with this. So this is the organic chemistry. Every undergraduate hates this class. I loved this class, hence why I do it as my job. I promise you guys, you have no idea what any of those things are, right? You're like, what is going on up there? The N is for nitrogen. Do we at least know that? Awesome. Okay, good. I knew you did. Okay, anyways. So, this was, do you remember me talking about the high throughput screening? Remember me talking about that? And we're gonna get a hit from the high throughput screening. So this was the hit that they got, this diazepam structure. Diazepam is this 7-membered ring here with 2 nitrogens in the 1 and the 4 position. So this is pretty cool. This was a really neat structure. And this got down to 630 nanomolar. That's what NM is, 630 nanomolar. This number, if you can't tell by looking at all the other structures, we want it to be really low, really low. If your number is really low, you have an amazing affinity for your target, what we call potency. So what they did first is, in terms of what I do on a daily basis, we're going to do lead optimization. This isn't the best compound. All right, we can do better. We are going to do better. So how are we going to do better? Well, they decided to change this on your guys, the left-hand side, and they did what this is a pyrimidine benzene ring here, and this got down to 147 nanomolar. So the only thing they changed with this left side here and got to 147 nanomolar. Not too bad, right? 3.5-fold better than the hit. So this was really exciting. They're not done that, though, guys. What they also decided to do was change the right-hand side, and they wanted to change what we call the substituents on this phenyl ring here. So they added just a phenyl up here at the top, got rid of this methoxy, and it got down to 19 nanomolar. Well, let me tell you, I would be massively excited about 19 nanomolar. Oh, they didn't stop there. They decided to do what this is. These are 3 nitrogens in a row. This is a triazole. So this is they also decided to put a triazole here and got down to 28 nanomolar. In terms of drug discovery world, these are pretty equal potent. They have an equal affinity for your target. Then they decided to, well, let's put this stick right here. You guys are like, what is a stick? It's called a methyl group. You won't understand that till you get to college. But that stick got it all the way down to 2 nanomolar. 2 nanomolar is amazing. So they decided to put these two together, and they made what they termed their lead compound. So this is very exciting. This was the optimization process that I was telling you guys about. They put all those facets together. They probably made 1 million libraries in terms, well, not 1 million, but I'm exaggerating, but a lot of libraries out here and also in terms of what was going on out there. And these were just the best ones that I pulled out for you guys. So this is the lead compound. Well, if you notice down here, they had some problems, right? We're going to be resilient. We're going to bounce back. They had really poor pharmacokinetics. That means how it's moving through your body. And they also had, the laser is just wanting to stay on. That's cool. They also had reactive metabolites. So that means when it was being metabolized, it was actually reacting and giving really poor toxins in your body, giving really awful side effects. And so they decided to move forward and really look at those metabolic hotspots. So in those metabolic hotspots, they had here, this benzene ring was a huge oxidation problem. The diazepine in the middle was getting metabolized and broken apart. And then you also had this benzene ring. So this methyl group wasn't really doing much to block the metabolism sites. So what they decided to do is, again, they changed this left-hand piece here, and they got, this is a benzoxazole, and they added a chloro in this position. And that chloro blocked any site of metabolism here. It wasn't being metabolized at all. They also kept this the same and only added a methyl group here. This caused a lot of steric hindrance in terms of what's going on here, and steric just means they were too close together. And so because the structure was really close together there, it ended up blocking this site of metabolism. So this is survorexant, and it got down to less than 1 nanomolar. They were very excited about this compound, and they were even more excited when they got to the preclinical studies because it was metabolically stable, like I just mentioned. It promoted sleep in every animal they ever tested it on. Rats, drugs, non-human primates, which are the monkeys. They did this every animal, it promoted sleep, which is great. So they moved on to the clinical trials. What was even greater is they got dose dependency, which is what I was talking about earlier. You're going to increase the dosage, you're going to increase infect, and they also improved sleep onset. So that's going to be, you're going to fall asleep faster, and you're going to maintain sleep just as well. And then it also increased total sleep time. So all of these insomnia patients were not only falling asleep faster and maintaining sleep, they were sleeping longer and feeling better the next day. It also, not at the higher doses, which I guess is kind of acceptable, at the lower dosages, people were able to drive the next day, which was amazing because that was their main goal in the beginning, right? They wanted to come up with a medicine, insomnia medication, that would allow somebody to drive the next day. I really like this e-card. I'm not a huge e-card person, but I found this online. It was really funny. And insomnia sharpens your math skills because you spend all night calculating how much sleep you'll get if you're able to fall asleep right now. So everybody, don't go to sleep tonight because you're going to sharpen your math skills by calculating it. I'm just kidding. Please sleep. Sleep is really, really good. I love sleep. Okay. What's just exciting about this example is I found out this was FDA approved just this August. So this is really exciting for this company. It's a new receptor, the orexin receptor. It's going to be the first one on the market, and I think it'll probably be able to go to patients full time in March of next year. So this is really exciting for this company. So how am I going to round this all out? How am I going to tell you guys? I told you got to be determined, you got to be resilient, you got to be unwavering. So how am I going to round this out? We want to finish with a great quote from two amazing ladies in the world, Miss Marie Curie and Dolly Parton. So first we'll start with Marie Curie. Do you guys know who she is? Oh, I love, man. Now that made my day. Not how loud anybody was earlier this morning. That made my day. She's a great lady. She was the first woman to win the Nobel Prize in chemistry. She also was the first woman to win 2 Nobel Prizes in chemistry and physics, and she was a mother of, if my memory serves me correctly, 4, and she was married to a scientist. Whew, this lady had competition, right? She had competition. But this is a great quote. I love it. We must have perseverance and, above all, confidence in ourselves. We must believe that we are gifted for something and that this thing must be attained. We have perseverance. You have to have confidence that you can do it. Whatever you set out to do, you have to be purposeful. You have to believe that you're gifted. Whether or not that's for STEM, I would love it to be in a STEM career. You may not be, right? Whatever you are gifted for, you must attain it. God gave you a gift, you need to obtain it. You need to be 2% genius and 98% hard work. I'm telling you, women work harder than any man, I guarantee you, ever. That's how we are, right? We have to work harder than. We are 2% genius, 98% hard work. 98% determined, resilient, and unwavering. Turn the me into you. Last thing I want to leave you with is find out who you are and do it on purpose. Miss Dolly Parton she's a fabulous, fabulous lady. She knows who she is, and she does it on purpose every single day. I hope you all can find out who you are. I hope it's a determined, resilient, unwavering girl, because I know I am, and I'm sure you can be too. Are there any questions? That's my little girl, and she's also back there. Say hi, Carrie. She's what I live for every day. Any questions, guys? Yes. Yeah, so like I said, I wanted to be a doctor. So I started out really shadowing a lot of doctors in high school because that's what I wanted to do. Obviously, that didn't end up being the case. So I'm not sure what kind of internships are available for high schoolers. I think Fernica did the Project Seed, which is great. If you guys really want to get into research, that's something that you should definitely look into. And the biggest thing that I would say is if you want to do research, do it right as your freshman year. I didn't start till I was a senior in college doing research. I started pretty late. So if you want to do research, or if you just want to see if you like it, talk to the professors your freshman year of college, no matter what college it is, whether it's MTSU or Vanderbilt, Notre Dame, USC, whatever. Talk to professors. They are more than willing to take students. A lot of times they have to take students, and they are going to teach you, especially being a freshman. They're not going to leave you in a lab and say, do this. They're going to help you. They're going to mentor you, and that's going to be great for you. So if you definitely want to pursue research, I would start that. Look into the Project Seed, and then definitely your freshman year of college, really look into doing undergraduate research right away. Any other questions? Yes. Well, that's actually a great question. So it hasn't been a problem for me to keep up with current technology. What's great about where I work is they're already up on the latest technology in terms of research. So we have the latest equipment. Where I work, we model a pharmaceutical company in an academic setting, which is really awesome. So we don't have all those politics when it comes to the pharmaceutical companies, with funding and things like that. But we have all of their abilities in terms of figuring out if a reaction worked or not. NMR. You guys heard of NMR? I guess she has. Doesn't like it. That's how you determine structure, if you made the right thing. So the company I work for kind of allowed me to step up into that. But that was a learning curve for sure, because at MTSU at the time, we didn't have a lot of those facilities. So we have a great NMR here at MTSU, which is great. But beyond that, some of our facilities, I think now with the new science building, they're better off. I haven't been back for a little while, right? So, but I think in terms of where you get a job, that's part of your job training. It's figuring out the technology. Does that answer your question? Okay, great, awesome. Anybody over here? Yes. So the name of like what I do. So my job title is research assistant, and that's because I haven't been there long enough in order to get into like the big, what they call drug discovery scientist yet, but I'm on the horizon to get there. But I'm a research assistant, but essentially what I do is medicinal chemistry is what it's really called. That's what I do on a daily basis. It's called medicinal chemistry. So it's organic chemistry in the medicine world, essentially is what that is. There was another hand. Yes. Oh, definitely. Oh, yeah. Yeah, we, actually my boss, Craig Lindsley, his niece has come and shadowed me and some of the others that I work with several times. And we have high school students. Actually, I think it's a project, it is project, I guess, in terms, in conjunction with Vanderbilt. I would talk to your high school chemistry teacher. We have high school students come around the lab a lot. We even have one, I think 2 or 3 last summer. I think it was a summer thing, a summer research thing. I don't know if you stay at Vanderbilt. That's something you should definitely look into if you really want to do research, is look into that, because that will look great on any sort of college application. But yeah, we definitely have people come around all the time to job shadow. Yeah. Yes. Vanderbilt? Center for Neuroscience Drug Discovery. We actually have a new facility now. We just moved to Cool Springs. So do you guys know where that mall is? Literally right across the street. No joke. We go there and eat lunch all the time. But yeah, so it's a new facility, so it's brand new. It's awesome. It's beautiful. It's great. We have all the latest stuff. So that's been really nice in terms of getting off of the campus. And with that, anyways. So, any other questions? Yes. What to name the drug? You know what's so funny is I was actually talking. So of course I talked to my boss and a lot of my other colleagues about this presentation. I didn't want to say anything wrong, and we were talking about who gets paid to do that. There's got to be somebody that gets paid to like name a drug. I have no idea. I really don't. I have not a clue. I'm sure somebody gets paid some big bones, probably more than I do, to do that. But they're creative, I guess, or they don't know how to spell is another thing. Either way. There are some really weird names, yes. But if you guys, after hearing this, look at any sort of commercial and hear what they're actually saying, really pay attention to what the drugs are actually saying, and the companies, and look up those companies, all of this stuff is online. Clinicaltrials.gov, you can look up any drug and it will tell you the results of a clinical trial. It's awesome. So I would definitely check that out if you're interested in this. Like, it's really cool. Yeah. Off topic. It is. So this picture was taken 4th of July, actually last year. It's one of my favorite pictures. We make a cake every year, the 4th of July cake. Huh? Gee, do you like it, Carrie? You like the cake? It's yummy. So great, so easy. Mm-hmm. Huh? I'll tell you the rest. It's family secret. What are you talking about? Okay. Are we good? Everybody good? You don't ask me any more questions? No? Like my favorite color, or I don't know. Just kidding. My favorite color is none of those colors up there. Let me tell you. No, it's lime green. Girl, woo! Okay. All right. I have really enjoyed talking to you guys. Thank you for listening. I really appreciate that. Thank you, guys.